Hello, guys. This is the lecture 2 of biotechnology, and in this lecture, we are going to discuss the topic how the piece of DNA replicate in the host cell. First of all, you all know that we alter the DNA, which consider a characterizable, which we need desirable character in it, and then we are introduce it in a host cell. Now come in this topic, we are discuss how this DNA replicate in the host cell. Most likely the piece of a DNA would not able to multiply itself in the progeny cell of organism, but when it integrated into a genome of a recipient, it may multiply, be inherited along with the host DNA. Okay. First of all, it is not a multiply in a progeny cell, but when it integrated, you all know about this word integrated. Integrated means sum up, जुड़ जाना और part बन जाना, like that word. Integrated into the genome of recipient. Integrated in the genome of a recipient, it may multiply, be inherited along with the host DNA, being inherited along with the host DNA. Okay, thus, or because the alien piece of DNA has become a part of chromosomes, which has the ability to replicate. Now, when जीनोम के साथ इंटीग्रेटेड हो गया हमारा एलियन डीएनए जर्नलिस्ट से फॉरेन डीएनए. Now, what will happen? This is because alien DNA become a part of chromosomes, and we know that chromosomes has a good ability to replicate. You all know about during mitosis, the number of chromosome became double. During माइटोसिस द क्रोमोसोम नंबर बिकेम डबल. सेल डिवीजन से पहले होती है, जीवन फेज में होता है जब हमारे क्रोमोसोम नंबर डबल हो जाते हैं. यू ऑल नो अबाउट दिस. इन 11th क्लास, आई think, yes, 11th class, we are discuss about it. We are discuss about it means we are learn in this. In chromosomes there is the specific DNA sequence. In chromosomes there are specific DNA sequence called origin of replications, which is responsible for initiating replication. Now this is if the question arises, what is the origin of replication? Uri. Oh, alright. Origin of replication, you say that in chromosomes there is the specific DNA sequence known as origin of replication, which is responsible for initiation the replication. Now due to this alien DNA or foreign DNA is linked with origin of replication, so that this piece of DNA can replicate and multiply itself in the host cell. This can also be called as cloning. In further lectures we are come about this cloning also, or making multiple identical copy of any template DNA. Okay, any template DNA. Cloning is the process in which we are making the identical copy of any template DNA. This is from NCERT. Generally the students do not focus on this, they are focused on the higher thing, but I think so small things in future make a great thing, make a large thing. और हम ये भी नहीं कह सकते कि नीट में नहीं आएगा. सो हमें ये थोड़ा बहुत आ जाना चाहिए, और थोड़ा सा ही है, कोई बड़ी चीज़ नहीं है इसमें. लेट्स फर्दर मूव. You all know about this. Again, I discuss in a short later. Discuss it. Now, पहले हमने क्या किया, एक ऑल्टर किया डीएनए जिसमें एक डिज़ायरेबल करेक्टर थे. फिर हमने उसको क्या कर दिया, इनिशिएट कर दिया होस्ट सेल में. इस प्रोसेस को हम जेनेटिक इंजीनियरिंग भी कहते हैं, और ये जो है हमारा प्रिंसिपल भी है हमारी बायोटेक्नोलॉजी का. अब आपने इसको होस्ट सेल में डाल दिया. अब हमें पता करना है कि अब ये कैसे हमारे होस्ट सेल में रेप्लिकेट कर रहा है, क्योंकि फर्दर वी आर गोइंग टू डिस्कस अबाउट द प्रोसेस ना अबाउट दिस, बट हम थोड़ा ओवरव्यू ले लेते हैं. अब क्या करता है, जैसे ही हमारा उसके अंदर चला जाता है, पहली बात ये प्रोजेनी सेल में नहीं करता, तब तक नहीं करता रेप्लिकेट जब तक वो जो है इंटीग्रेटेड, मतलब इंटीग्रेटेड नहीं हो जाए जीनोम के साथ। जब वो इंटीग्रेट हो जाता है जीनोम के साथ, रेसिपिटेंट के जीनोम के साथ, तब क्या होता है, वो मल्टीप्लाई भी होता है, इनहेरिटेड भी होता है। क्यों? क्योंकि इसके वजह से वो डीएनए जो है पार्ट बन जाता है क्रोमोसोम्स का। क्रोमोसोम्स का पार्ट बनने का मतलब है कि उसमें भी एबिलिटी है मल्टीप्लाई करने की। फिर हमें पता है क्रोमोसोम्स में कुछ स्पेसिफिक सीक्वेंस होते हैं जिसको ओरिजिन ऑफ रेप्लिकेशन कहते हैं, विच इज़ रिस्पॉन्सिबल फॉर इनिशिएशन ऑफ रेप्लिकेशन। तभी तो हम एलियन डीएनए और फॉरेन डीएनए को इस ओरिजिन ऑफ रेप्लिकेशन के साथ मिला देते हैं। एंड मल्टी देन दिस डीएनए it multiply it in a host cell now. That's why this process is known as cloning. This overall process is known as cloning. That's it. Now further moving. First recombinant DNA. If question arise, who discovered the first recombinant DNA? That is Cohen and Bohr. First of all, we discuss the steps, then we come forwards to this diagram. This is the diagram. Now, let's further move. Stanley Cohen Herbert Boyer got the idea of linkage a DNA encoding for antibiotic resistance with a native plasmid of Salmonella typhi or typhirium medium. Okay, plasmid of bacteria. Let's further move. First step, isolations the antibiotic resistance gene by cutting out a piece of DNA from a plasmid, which was responsible for conferring antibiotic resistance. The cutting of DNA is done with the help of restriction enzyme, commonly known as molecular scissors. Okay, in first step, what they say, isolation. आइसोलेशन हो रहा है, आइसोलेशन द एंटीबायोटिक रेजिस्टेंस चेन बाय कटिंग आउट द पीस ऑफ डीएनए। किसके वजह से? बाय कटिंग आउट द पीस ऑफ डीएनए फ्रॉम अ प्लाज्मिड विच वॉज रिस्पॉन्सिबल फॉर कन्फरिंग एंटीबायोटिक रेजिस्टेंस। द कटिंग ऑफ डीएनए इज डन विद द हेल्प ऑफ रिस्ट्रिक्शन एंजाइम कॉमनली नोन एज मॉलिक्यूलर सीजर। मॉलिक्यूलर सीजर। Okay, further moving. The cut piece of DNA was linked with a plasmid DNA. This plasmid DNA acts as a vector to transfer the piece of DNA attached to it. Now, the cut piece of DNA was linked with a plasmid. The plasmid act as a vector which transfer the piece of DNA attached to it. DNA ligase enzyme help in linking the fragment of DNA with the vector. This make a new combinant circular autonomously replicating DNA create in vitro. It is known as recombinant DNA. When the newly formed DNA was transferred to Bacterium E. coli, it was able to replicate in a host cell by using a DNA polymerase enzyme. It was able to replicate in the host cell using DNA polymerase enzyme. If the question arises sometime, with the help of which enzyme E. coli replicate itself in the host cell, that is the DNA polymerase enzyme. The ability to multiply the copy of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. There are three basic steps in the genetically modification organism. And this I talk further. First of all, I explain or describe this diagram. This is the DNA of Salmonella typhi or typhirium, and this is the vector, that is DNA plasmid. Vector हमने पढ़ा ना, vector DNA है plasmid is generally act as a vector because it is help in the transfer of this particular DNA or piece of DNA or alien DNA we say further. Now this DNA is here, resistance towards antibiotics, then we are this DNA are we integrated it with a plasmid, and then recombinant DNA is formed having a desirable gene. Then we are put this in E. coli, and E. coli, when this is come in the contact with the genome, then it get ability of replicate, because it became a part of chromosomes, and hence we know that the chromosome has a great capacity of a multiply. That's why in the future E. coli this DNA has come, and this process is known as cloning. In further, there is the NCERT diagram also, which I discuss later on, because I discuss it in this, because this is given in the NCERT, and I think the diagram is must, because we know that with the help of diagram we can easily understand the things, and थ्योरी तो लिखना बहुत इजी होता है अगर हमें ये चीज़ आती है, तो थ्योरी तो कोई भी बना देता है, तो आपको डायग्राम दिमाग में होना चाहिए, थ्योरी अपने आप बन जाती है. आई suggest that. Now, there are three basic steps in genetically modifying organism. First of all, थ्री जर्नल स्टेप बोला है मॉडिफाई, अगर हमें मॉडिफाई करना है ऑर्गेनिजम्स को. फर्स्ट, आइडेंटिफिकेशन ऑफ डीएनए विद अ डिजायरेबल जीन. ऐसा नहीं कि कोई भी डीएनए निकाल दिया भाई, हमने तो बनाना है कोई अच्छा सा इंसान, या मान लो कोई अच्छे से आपने क्रिएशन करनी थी, पर आपको तो आपने ले लिया डीएनए गलत, तो ये तो नुकसान है. फर्स्ट ऑफ ऑल, फिर इसका मतलब ये है कि हमें आइडेंटिफिकेशन करनी है डिज़ायरेबल जीन की. ऐसे नहीं कोई हर कोई डीएनए का वो ले लिया हमने, कोई गंदा ही उसमें करेक्टर होएगा और उस करेक्टर को ही सोचते आगे करते हैं. नो, हमें डिज़ायरेबल करेक्टर लेना है. इंट्रोड्यूस ऑफ द आइडेंटिकल डीएनए इनटू होस्ट. इंट्रोडक्शन ऑफ आइडेंटिकल डीएनए इनटू होस्ट. Okay, maintenance of introduced DNA with the host and transfer of DNA to its progeny. This is the three basic steps. Okay, I, if you like, please like and subscribe my channel, and in further upcoming video I discuss the tool of recombinant DNA technology, that is restriction enzyme, or in which we are come across the word scissor, no, molecular scissor. It is generally the enzyme, restriction enzyme, known as endonucleases. एंडोन्यूक्लियसेस, और हम जो है एक ही रिस्ट्रिक्शन एंजाइम यूज़ करेंगे, इसको भी कट करने के लिए और इसको भी कट करने के लिए. आई एम डिस्कस इट फर्दर. सो इफ़ यू लाइक माई चैनल, प्लीज़ subscribe it, share it, and I hope you do that. Thank you. All the best.